Good morning, children. Welcome back to biology class. Sexual reproduction in flowering plants, part 8. Sexual reproduction events are grouped into 3 stages, namely pre-fertilization, fertilization, and post-fertilization events. Pre-fertilization events are gametogenesis and gamete transfer, formation of male and female reproductive structures in flowering plants. Pollen grains and pistil are the male and female reproductive structures. Gamete transfer is the pollination. Okay. Last classes we have discussed about the structure of the pollen grains, pistils, how the pollination will take place. All this we discussed. Then we discussed the second event in the sexual reproduction is fertilization. Okay, the fertilization, how it was? After entering one of the synergids, the pollen tube releases the 2 male gametes into the cytoplasm of the synergid. One of the male gamete moves towards the egg cell and fuses with the egg's nucleus, thus completing the syngamy. After the results, the diploid cell zygote is forming. The other male gamete moves towards the polar nuclei located in the central cell and fuses with them to produce a triploid primary endosperm nucleus. As this involves the fusion of 3 haploid nucleus, is termed triple fusion, since 2 types of fusions, which are the syngamy and triple fusion, takes place in an embryo sac. The phenomenon is termed double fertilization. That is the second event in the sexual reproduction. It's a common process in the flowering plants. The central cell after the triple fusion becomes the primary endosperm cell and develop into the endosperm, while the zygote develops into an embryo. Then today we are going to discuss about the third sexual reproduction event, that is the post-fertilization. Following the double fertilization, events of endosperm and embryo development, maturation of ovules into seeds, and the ovary into fruit, collectively termed as post-fertilization events. Now we'll discuss about the endosperm. Endosperm development continuing with the embryo development. The primary endosperm cell divides continuously and forms a triploid endosperm tissue. The cells of this tissue are filled with the reserve food materials. That is what is the function of that, and are used for the nutrition of the developing embryo. See, listen, the endosperm tissue contains the reserve food materials that are used for the nutrition of the developing embryo. In the most common type of endosperm development, the primary endosperm nucleus undergoes successive nuclear divisions to give rise to free nuclei. That means this stage of endosperm development, there is no cell wall formation, so they are called the free nuclear endosperm. After that, they are forming the cell wall formation occurs and the endosperms becomes cellular. The number of free nuclei formed before cellularization varies greatly. Now, we will take one example, the coconut. The tender coconut is a free nuclear endosperm because it contains more water in that. There the nuclei made up, many thousands of nuclei is present. There is no cell walls in the beginning. When the coconut becomes mature, white kernel is formed with the cellular endosperm. So that is the first in the beginning stage, that are the free nuclear endosperm, and when it is become mature, it is the cellular endosperm. Endosperm may either be completely consumed by the developing embryo, that are the non-albuminous seed, or the endosperm may be half consumed by the developing embryo, that is the albuminous seed during the seed germination, that we'll discuss later. Now, endosperm is the nutritive tissue. Now, next, the embryo development in the stage post-fertilization events. Embryo develops at the micropylar end of the embryo sac where the zygote is situated. Most zygotes divide only after certain amount of endosperm is formed. This is an adaptation to provide assured nutrition to the developing embryo. Though the seeds differ greatly in the earlier stages of embryo development is called embryogeny, are similar in both the monocotyledons and the dicotyledons. The stages of embryogeny or the embryo development in a dicotyledons embryo we'll discuss now is the zygote give rise to the proembryo. In the beginning stage, it is the proembryo, and subsequently to the globular embryo, then heart-shaped embryo, then the mature embryo. A typical dicotyledonous embryo consists of an embryonal axis and two cotyledons. Last year you have studied this all. The portion of the embryonal axis is the above the level of cotyledons is the epicotyle, which terminates with the plumule, and then later it is the stem portion. The cylindrical portion below the level of cotyledons is the hypocotyle that terminates at its lower end in the radicle or root tip. The root tip is covered with a root cap. Okay, a typical dicotyledonous embryo consists of two parts. One is above the soil and one is half portion below the soil. But middle one embryonic axis is there. Embryonal axis and cotyledons, that are the two cotyledons. The portion of the embryonal axis above the level of cotyledons is the epicotyle, that is becoming the plumule and also the stem tip. and the below the cotyledons it is the hypocotyl that is becoming the radicle or the root tip covered with the root cap. Embryos of monocotyledon seed. Okay, monocotyledon's embryo possess only one cotyledon. In the grass family, the cotyledon is called scutellum, that is situated towards the lateral side of the embryonal axis. At its lower end, the embryonal axis has the radicle and root cap enclosed in an undifferentiated sheath is called coelorhiza, that is becoming the root. The portion of the embryonal axis above the level of the attachment of scutellum is the epicotyle. Epicotyle has a shoot apex and few leaf primordia enclosed in a hollow folia structure is called coeloptile. That is above the soil it is present. So that the grass family or the monocotyledons portion contains the one cotyledon, and the reserve food contains it is called scutellum situated towards the one side, then the embryonal axis. Below the soil it is coelorhiza, and above the soil it is the coeloptile. Thank you.